Bismillahirrahmanirrahim. Alhamdulillahirabbil alamin. Wassalatu wassalamu ala asyrofil anbiya'i wal mursalin wa alaihi washabihi ajmain amma ba'du. Assalamualaikum warahmatullahi wabarakatuh. Di pagi hari yang berbahagia ini kita akan mendengarkan online lecture exchange yang akan dibawakan oleh Associate Professor Muhammad Arifin bin Kaderi PhD dengan judul Application of Cultured Cell in Genetic Engineering and Islamic Perspective. Perlu saya sampaikan terlebih dahulu untuk susunan acara pada hari ini adalah yang pertama nanti kita akan ada pembukaan dengan bacaan kalam ilahi oleh Prima Rizky. Kemudian nanti akan disusul oleh sambutan dekan, dan kemudian kita akan masuk ke acara inti, yaitu kuliah tamu oleh Associate Professor Muhammad Arifin bin Kaderi dari International Islamic University Malaysia, dan dilanjutkan dengan Q&A. Untuk menyingkat waktu, Prima sudah masuk ke ruangan, silahkan untuk membuka dengan kalam ilahi. Tes. Silakan Prima.
Baik Bu, terima kasih. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. A'udzu billahi minasy syaithanir rajim. Bismillahirrahmanirrahim. Qad aflahal mu'minun alladzina hum fi salatihim khashi'un walladzina hum 'anil laghwi mu'ridun walladzina hum lizzakati fa'ilun والذين هم لفروجهم حافظون إلا على أزواجهم أو ما ملكت أيمانهم فإنهم غير ملومين فمن ابتغى وراء ذلك فأولئك هم العادون والذين هم لأماناتهم وعهدهم راعون والذين هم على صلواتهم يحافظون أولئك هم الوارثون الذين يرثون الفردوس هم فيها خالدون ولقد خلقنا الإنسان من سلالة من طين ثم جعلناه نطفة في قرار مكين ثم خلقنا النطفة علقة فخلقنا العلقة مضغة فخلقنا المضغة عظاما فكسونا العظام لحما ثم أنشأناه خلقا آخر فتبارك الله أحسن الخالقين ثم إنكم بعد ذلك لميتون ثم إنكم يوم القيامة تبعثون ولقد خلقنا فوقكم سبع طرائق وما كنا عن الخلق غافلين صدق الله العظيم. السلام عليكم ورحمة الله وبركاته.
Alhamdulillahirrahmanirrahim. Selanjutnya untuk sambutan dekan, kami mengundang Bapak Dekan Fakultas Kedokteran Unisura, Dr. Setio Trisnadi SHS PKF untuk memberikan sambutan pembukaan. Waktu dan tempat kami persilakan.
Terima kasih Bu Putri. Assalamualaikum warahmatullahi wabarakatuh. Innalhamdulillah nahmaduhu wa nasta'inu wa nastaghfiruh wa na'udzu billahi min syururi anfusina wa min sayyiati a'malina may yahdihillahu fala mudilla lahu wa may yudlilhu fala hadiyalah. Asyhadu an la ilaha illallah wa asyhadu anna Muhammadan abduhu wa rasuluh la nabiyya ba'da. Amma ba'du. The Honorable Professor Muhammad Arifin bin Kaderi PhD, lecturer from International Islamic University Malaysia, who will deliver a lecture on the topic aplikasi kultur sel dalam genetic engineering dalam perspektif Islam. Respectable all participants, General Lecturer Medical Faculty of Universitas Islam Sultan Agung. Dear ladies and gentlemen, first of all, let's pray and thank unto our God, Allah Subhanahu wa Ta'ala, the Lord of this world, who has been giving us mercies and blessing that we cannot count. So we can turn and gather in this unforgettable place in a good condition and happy situation. Secondly, my salawat and salam always be with our best Prophet Muhammad Sallallahu Alaihi Wasallam, who has guided us to know and do the right thing to life in this world, namely Islam, the religion which guides mercy to all world. Thirdly, I would like to say thank you very much to Professor Muhammad Arifin bin Kaderi, PhD, for the excellent cooperation between IIUM and MISULA, so that they can share knowledge for the advancement of medicine and the benefit of the people. We hope that this collaboration can be improved in research and community services. Thank you. Have a good general lecture. Assalamualaikum warahmatullahi wabarakatuh. Thank you, Master of Ceremony.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Pak Dekan. Selanjutnya, waktu akan saya berikan ke moderator untuk hari ini. Moderator for this general lecture is Dr. Fikri Taufik, MSc Med PhD. He will be chairing the session for our general lecture. But before we start, can you please all activate your video, because we would like to take your picture before the general lecture. So please, if you can, please turn on your video. There are 4 pages on this Zoom room. So, okay. Kita siap-siap ya, foto ini untuk halaman pertama yang terlihat di screen saya. Siap. Oke. 1, 2, 3. Okay. Second screen. Okay. Second screen. Ada yang masih off second screen, sebentar. 1, 2, 3. Ya. Third screen. 1, 2, 3. Screen terakhir 1, 2, 3. Okay. We can proceed for Dr. Fikri. The time is yours.
Thank you very much, Ms. Putri. Okay, good morning, ladies and gentlemen. Today we have online lecture exchange. The topic is about application of culture cell in genetic engineering and Islamic perspective. And we have Associate Professor Muhammad Arifin bin Kaderi, PhD. Good morning, Mr. Arifin. Can you hear my voice?
Yes, Assalamualaikum warahmatullahi wabarakatuh.
Okay, Waalaikumsalam warahmatullahi wabarakatuh. Good morning, Mr. Arifin. Before we start this lecture, I think it's better to know each other. So let me introduce myself. My name is Fikri Taufik. I am belong to Department of Physiology since 2011 until now. But during 9 years, I focus on study, like master course and PhD course. And I just finished my PhD course in this year on March. Okay. And
congratulations. Congratulations on your PhD.
Okay, thank you, Mr. Arifin. And also, I'd like to introduce Mr. Arifin to all of the audience. Maybe I will share the screens about the curriculum of Mr. Arifin. Okay, we have Mr. Muhammad Arifin bin Kaderi, PhD. He belong to Department of Biomedical Science, Kuliah of Allied Health Science, International Islamic University, Malaysia. And his current position is assistant professor, and his nationality is Malaysia. Let's go to the next slide. Yes, his work experience is very great. From postdoctoral research fellow 2012 until 2014, and then become the head of research, Kuliah of Allied Health Science, International Islamic University, Malaysia, from 2015 to 2018. And then become the editor of International Journal of Allied Health Science from January 2011 until now. And then become the assistant professor on Department of Biomedical Science, Kuliah of Allied Health Science, International Islamic University Malaysia from 2010 until present. And then associate professor on Department of Biomedical Science, Kuliah of Allied Health Science, International Islamic University from 2020 until present. And then his academic qualifications, he took a PhD in biomedical science in International University of Malaysia 2000, and then master course on MSc in University Science Malaysia 2004, and then PhD in clinical genetics, Uppsala University, Sweden, 2010. It's a very great academic qualification, Mr. Arifin. And then many award and recognitions, and it's very great. And then the last about the publication highlights. There are many, and this is just part because I already read his curriculum vitae. The complete curriculum vitae is many publication highlighted. And I think it's enough for introductions, and I think it's better to not far these introductions. Okay, Mr. Arifin, please can give the lecture. Time is yours.
Thank you very much, the moderator. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Bismillahirrahmanirrahim. Alhamdulillahirabbil alamin. Wassalatu wassalamu ala asyrafil anbiya wa mursalin wa ala alihi wa sahbihi ajmain. Rabbi shrah li sadri wa yassir li amri wa hlul 'uqdatam min lisani yafqahu qawli. Subhanaka la ilma lana illa ma 'allamtana innaka antal alimul hakim. Wala haula wala quwwata illa billahil aliyyil azim. La haula wala quwwata illa billahil aliyyil azim. Wala haula wala quwwata illa billahil aliyyil azim. Thank you very much again. Thank you very much, Unisula, for inviting me for this event. I think I'm very glad to be together with the fellow academician and also students from Indonesia. It's actually my, I think, third communication online with Indonesian University. Recently, I have actually. Before that, sorry, I have to address the dean for Faculty of Medicine, Unisula University, Universitas Islam Sultan Agong, ya, yes, sebutannya. Bapak Setio Trisnadi, as the for giving the support and permission actually for this event to occur, for us to be together. Thank you very much also Ibu Putri Yuningtias for our communication and helping me to get familiar with Unisula in general, and also preparing myself for this event today. Thank you very much for all the communications. Also, our moderator today, Dr. Fikri, for thank you very much for introducing me to the audience.
You're welcome, Mr. Arifin.
My fellow professors from Unisula, associate professors, academicians, students, medical students, and also students from other faculties. Hope, thank you very much for having me. Thank you very much for your time. for today's sharing session. All right. Um, I think I'll try to share my screen. Yeah, this one, yeah. Is it possible? Is it okay? Oh, no.
It's not showing yet, Doctor. Maybe.
Yeah.
Uh,
I'm actually not very familiar with Zoom. Usually in my faculty, we use Google Meet. So yeah, I'm still have to learn. Where is it that I should?
You can press the share screen button, the green one,
all right.
the green one under, and then you can locate your file, the one that you are going to use for your presentation.
Okay.
Have you found it?
Okay. All right. I'll. What is it? I have to put it in. Is it? Do I have to put the file in Google Drive or because?
Oh no. You have to open your file first. You open your presentation file first, and then you press the share screen button.
Okay.
There's a share screen button, the green button. You press that, and then there's a tray that locating your open file. Okay.
Okay. All right.
Okay, it's already.
So can everyone see my slide now?
Yes.
Okay.
Yes.
All right. Okay. Thank you very much. Yeah. Okay. So as I said, this is actually my third communication with Indonesian University. Before this, I've been in IPB, Institute Pertanian Bogor, in 2018 for research fellowship. So I have my teachers there in IPB. So and then we actually planning for collaboration in research, especially in canine distemper virus, which is a virus that affecting our tiger population in Malaysia. So we are communicating back and forth, planning for the research. At the same time, since I've been given by the university to look after the cultural part to develop the system for our students, building up efforts towards fusion between culture and research and science. So I also learn and communicating with other university, UNES, I think in Semarang also, right? And also USU, University Sumatera Utara. So yeah, so also yeah, I think I'm fond and I have a very good experience, all pleasant experience working together with universities in Indonesia. I'm very glad to build up collaboration with our fellow academicians, especially in Sumatra and also in Java Island. So, I think yeah. Okay. All right. So the topic that has been given to me today is application on cultured cell, the genetic engineering and Islamic perspective. First of all, I would like to say that this is I don't consider this as teaching. So I consider this as more of sharing. I believe and I'm sure there are many professors and also academician researchers in Unisula that actually has experience, vast experience, I think more experience than me in this field. So I think I'll just share, and I would also like to take this opportunity to learn from each other, so that we can advance together, inshallah. Yeah. So as Dr. Fikri mentioned earlier today, yeah, I will not go in detail in this because this has been mentioned by Dr. Fikri earlier. Just want to highlight one, which is my work or my collaboration with IPB in 2018 under the research fellowship, the Southeast Asian One Health University Network. This work we actually doing we did some research on coronavirus actually during that time before the time of COVID. We actually expected some uh, some pandemic or for some new cases that would occur due to coronavirus during that time, but we don't know which one. Um, we hope that with the knowledge we would help the country to how to respond with the emerging pandemic threat. And um, yeah, we didn't expect. We were actually worried the pandemic will start from certain part of Indonesia, but alhamdulillah that did not happen. It starts from China. So yeah, it's not the worst that we the expect. And what even though at the moment we all are facing some problem, we are facing this situation with COVID-19. But in a way, I think we are glad that we did some research in this field so that we were more prepared than and we have some background knowledge on coronavirus before the virus pandemic occurs. So Alhamdulillah, for example, like in Malaysia, our knowledge has been very useful for the country to respond and to strategize on how to respond on the COVID-19 pandemic that we are facing today. So Alhamdulillah. So in a way, I'm also consider myself a student of university in Indonesia because I learned a lot from my teachers and my fellow scientists in Institute Pertanian Bogor. And I hope, inshallah, in the future, I can also learn from great and wonderful academics and researchers in Unisula, inshallah. Yeah, so start with introduction to in vitro culture, right? And what is in vitro culture? I think many of you already know about in vitro culture, but I think for some of the students who feel that this is new to them, you know, let us start with the definition. As defined in the the online medical dictionary, in vitro culture is defined as the propagation of living tissue cells in specific media which is conducive for the cell's growth, right? And in the American Heritage Medical Dictionary, in vitro culture is defined as the technique or process of maintaining or cultivating cell. I cannot see actually cell or tissues derived from a living organism in a culture medium, right? A culture cell or tissue grown by this technique or process is called tissue or cell culture, right? There are several types. Beside the cell culture, there are also different type of culture. Number one, the organ culture. I have to admit that I'm not very familiar with organ culture. I've never done organ culture in my career so far. All right. So in organ culture, when we refer to mammalian organ culture, It refers to the culture or cultivation of the entire embryos. In most cases, the component that is being used for this culture is the embryo, or in some cases, the organs that are excised or cut from or separated from the body and culture, right? There's some advantage as you can see in the figure on the right of on the right side of the slide. Example that has been given is the culture of the whole kidney. In this case, is the mouse kidney, right? So it's actually the whole organ that is cultured, right? And in this example, for the study of vertebrae, right? The entire some part of the vertebrae itself, the backbone of the mouse, is separated from the animal and then cultured in the laboratory in vitro, right? And there are some advantages of doing tissue culture. Number one, taking the whole organ means that we take the all of the cells or all of the tissues that are required for the maintenance of the organ, for the physiological function of the organs. All right. So the normal physiological function are supposed to be maintained by the organ culture technology. All right. And the cells in the organ would remain to be fully differentiated. The normal process of mitosis and maintenance of the organs are supposed to be maintained, right, or to be preserved in the organ, even though it's just on for a limited time, right? And there are also this advantage of this technique. Of course, number one is the difficulty in getting the organ or the culture itself to be successful or to be function or to be able to maintain it over a certain lengthy period of time, right? One of these advantages is scale up is not recommended, so we cannot doing it in high throughput or in many numbers of the organs, right? So because we also play with the bioethics and so on when we work with laboratory animals, so we have to always try to, as much as possible, limit the number of the animal that being used for this kind of work, right? You cannot use just infinite number of the subjects. And of course, in this case, usually it's the organs from the laboratory animal that is being used for the organ culture. Right? So far, I've to my knowledge, there have not been like the whole culture of human organ done in the laboratory so far, right? Also, the growth of the cell is usually slow, because when you take an organ from the individual, from the normal condition of the animal, and put it in the lab, so the growth of the organ will be very slow, and fresh explantation is required for every experiment, because of course, we cannot maintain the organ over a lengthy period of time. So we can only do the organ culture in the lab and then do some experiment. But if you want to do it over a lengthy period of time or over 6 months, we all, of course, we need several explantation, many repeated organ culture. So as shown here, it's up to 21 days only. All right. So it won't be long lasting. All right. Like not like cell culture. All right. Apart from organ culture, this is another technology which is at a smaller level is tissue culture. Right. Tissue culture, of course, it's very popular in plant science. Right. We can the technology now allow to call it grow plant continuously without using the seed, but instead using the tissue culture. That's very common and very popular in plant science. But in mammalian cell also that is possible. Right? There are also work that's been done on tissue culture. In fact, I think in IIUM, in our university here in Kuantan, Pahang, Malaysia, we have a group of researchers who are very, very active in tissue culture and tissue engineering, especially work on cartilage, right? Culturing cartilage for orthopedics application, right? In tissue culture, fragments of excised tissue are grown in culture media. Instead of organ, we take some part of the organ and the tissue, and we grow in vitro in the laboratory. All right, there are some advantages of doing this. Yeah, so some of the advantages is some of the normal function of the cells, or sorry, the tissue is maintained, because when we take the tissue, we have the cell that we want to study, but also at the same time we have all the cells that provide the microenvironment for the target cells that we are studying. So it provides sort of like a normal physiologic condition for the cells, right? Because the cells communicate with each other. So some of the supposed normal function in the actual in vivo condition is maintained, right? And it is better. It's believed to be better than organ culture for scale up, but not ideal. All right. Yeah. So still we are quite limited in terms of how much we can do with the tissue culture. All right. And the disadvantages is one of the advantages that usually discussed in the tissue culture community is the whether the original organization of tissue is actually representative of the condition in vivo. That is, I think, being discussed in the tissue culture community. I myself, I must admit, I've not done much of the tissue culture. It's I think different level of expertise and much more difficult than cell culture, to me at least. All right. So my experience in tissue culture is actually very limited. I must admit that. And come to the topic of the day, which is tissue culture. All right. And to do this tissue culture, to do the cell culture, the tissue from an explant, right, from the organ or the whatever that we dissect from the animal or from the human individuals, we disperse. Okay, we separate them, separate the cells, usually by using enzymes, enzymatic technology, right? And we put them into a cell suspension, which may then be cultured as monolayer or suspension culture, right? So the most popular way of cell culture is, of course, the monolayer culture, in which the cell will grow at the bottom of the petri dish, right? But we can also do suspension culture, and there are actually so many um, variation of how we can manipulate the cell culture. Some, I think, in IPB, there's one group in Institute Tanambogo. There's one group that is very experts in 3D culture, right? They call it three-dimensional culture of breast cancer cells. They are, I think, very good in doing that. Even I think universities from all over the world collaborate and learn from them. And we also have some collaboration with them to do the 3D cell culture in IPB. All right. So the advantages for cell culture, we can develop a cell line over several generations. You know, we can keep or we can maintain the cell line for many years. All right, depending on what type of the cells that we're using. All right, that we are studying, and scaling up of the cell culture is of course possible. We can do it. Making it in bigger volume, and then we can even share the cell lines with other laboratories in the world. If we have if we establish certain new cell line, for example, right, we can share it with we can scale it up, and then we can share it with our collaborators, for example, right. But there are of course issues with regard to cell culture. One of the disadvantage that being discussed and of concern with the researchers is that the cells itself may lose some differentiated characteristic. It may change over time. The character of the cells would change. For example, one of the very popular example is actually one of the earliest cell culture or cell line, which is HeLa cell. I think many of you, I think, have heard this name of the cell, HeLa, which is cell line that is taken originally from cervical cancer cell. There's even a movie, new movie with regard to HeLa cell. With regard to HeLa, HeLa stands for Henrietta Lacks, which is the name of the individual that from which the cell line is taken originally. All right. So it's very well known now that the HeLa cell that we have actually we cannot even say that it is a human cell, because it has mutated because it's already been used for so long, so many years. So many mutation has been occurred, and it's now in some laboratories it doesn't have diploid. I mean, human cells must have diploid chromosomes, right? Number of chromosomes should be 46 chromosomes, and then but in HeLa cell it may have double, and a lot of aberration has been observed in HeLa cell. And then very common that HeLa cell infused with other type of cells in the laboratory. So if you have a HeLa cell in a laboratory, be very careful if you work with other cells, because I've been working with nasopharyngeal carcinoma cell, all right, cell line. Nasopharyngeal carcinoma is the carcinoma of the back of the throat, right? So we do have cell line for nasopharyngeal carcinoma, or NPC, but there was one study, or I think a couple of studies has been done on NPC cell all over the world. All right, all over the world. We want to standardize, want to check the quality of the NPC cell line in many laboratories in the world, including the ones in Harvard and in Oxford. But what we found is all of the NPC cell line in the world has been contaminated with HeLa cell. So if we have a HeLa cell in the laboratory and we grow it together, somehow, if someone is not careful in doing the culturing or doing the passage, for example, contamination of HeLa, the HeLa cell can actually dominate or can infuse with the other cell lines. So so far, there's no NPC cell line that is actually pure NPC. That's very sad, very difficult, making it very difficult for us to study NPC nowadays because because of this issue, right? So just to say that doing cell line, there are some issues with regard to some of the cells may lose the characteristic of the original cell. So even though we know, for example, we are studying a cancer cells, which is actually not normal, but the cell that has been grown with, and especially cell that has undergone many passage, many round of culturing, may have changed. It's not the same as the cells that is taken from the tissue originally. It's not representative of the cells of the tissue in vivo, right? There is some issues that is being commonly discussed, right? Oh yeah, before I continue, may I use? I think the audience are all of you are Indonesian and understand sort of Malaysian Indonesian language, right? So can I mix between English and Malaysian and Indonesian, insyaAllah?
Yes, of course, Mr. Arifin, no problem, because we negara serumpun, that's why we can understand Malaysian language and also Indonesian language. Please, Mr. Arifin.
All right, thank you very much. So sometime bila cakap di bahasa Inggeris berterusan, kadang-kadang kita juga lupa perkataan-perkataan hilang dari memory. So if I can use sometime Malay language or Indonesian language, make it more continuous, more effective for the communication, insyaAllah. Thank you, Mr. Moderator. Right. So it's just summary, right? Summary of different type of culture, right? So the type of culture, there are different modes of culture that are represented from in this figure from left to right. The first is an organ culture on a filter disc on a triangular stainless steel grid over a well of medium, which is seen here. The organ is put under the stainless steel grid and grown in the culture medium. All right. The second is explant for cultures in the flask with section below, all right, and with enlarged detail in a section in the lower diagram, right? So this is a tissue grown in vitro, and we expect that the tissue will grow sideways, right, and cover the culture medium, right? Okay, and showing the explant and radial outgrowth under the arrow. So the third vessel, right? This is, I think, many of us have seen this flask, right? The third vessel with enzymatic disaggregation generating a cell suspension seeded as a monolayer, right, in the lower diagram, right? And then the fourth is a filter well showing an array of cells. This is organotypic culture and seen in section in the lower diagram combined with matrix and stromal cell. This sort of like combination, trying to mimic the condition in vivo in which we provide also the matrix and a stromal cell to support the growth of the cells that we target, right? So we're providing sort of like communication. We try to mimic the condition in the body So we put the matrix and also the stromal cell for the cells that we grow. I think this is also being done very commonly, very widely in the laboratory in IPB. I think it was the only lab in Southeast Asia that specifically grown primate cell, because the laboratory that I work in is the primate center, primate research center in IPB, which is actually the only what do you call it, certified, globally certified primate research center in Southeast Asia. I think yeah, very good laboratory in Indonesia. I think even in Malaysia we don't have the specified, very specific research center for non-human primate. Yeah. Okay, why do we need cell culture? All right. The main reason why we do cell culture, especially in the field of research, is to overcome problems in studying cellular behavior. All right. So a limitation of studying in vivo. All right. Such as the confounding effect of the surrounding tissues, right, and variation that might arise in animals under experimental stress. So in earlier generation of researchers, especially biological science researchers, the use of animals, of course, is very common. I think it's still very common in our laboratories around the world today. But of course, there are some limitations, because when we work with laboratory animals, the animals can be affected by the stress, right? And in many laboratories, also work in auditing some of the laboratory animals in our university, and also in some of the universities in Malaysia, and we see that there are some practice that not very proper for research. For example, in many of the research laboratories, they keep the mice with rat together in the same laboratory, which is actually very bad, because when we are studying the mice, actually mice is very affected. In fact, rat can actually be the predator of mice in normal conditions. So mice will be very stressed. If we put mice together with rat, they will be very stressed, and actually change. It may give wrong result. Actually, it does not represent the actual physiological condition of the mice. Right. Same thing with if we keep in some worse laboratory, we have like snake together with other rodents, which is very bad. All right. So that is the one of the issue of, and also when we keep the mice, for example, the New Zealand or rabbit, for example, the New Zealand white, which is actually the animal that is not native for our condition in tropical countries. So we use rabbit, which is not from our localities, and we study them. So the issue is, does the environment, the tropical environment, changes the behavior of the rabbit and the physiological condition of the rabbit, for example? That is also an issue. All right. And also confounding effects of surrounding tissues that may interfere with the result of the study or the experiment. All right. And also, for example, now there are more and more issues with regard to the use of animal for research, in which the animal ethics committee has been set in many universities. I think it's supposed to be set in all of the universities, like in Malaysia. So of course, always the emphasis is in reduction of the number of mice, and if there are alternative way of doing the study without using laboratory animal, that should be done. So and again and in addition to that, there are so many groups of non-governmental groups, the animal right group that oppose to the use of animal in the laboratories, and sometimes it can be quite dangerous. For example, like the lab that I use in the institute that I used to work in Sweden, they even hide that those people who do not work with laboratory animal, we actually do not know where is the laboratory the animal laboratory lab, right? Because there are cases in Stockholm in Sweden where the animal lab has been bombed by the animal right extremists. To that level, they have the animal right extremists who attack the animal laboratories. So it can be quite dangerous. So we always want to reduce the number of animal for any study. So if we have any alternative, we should use that alternative. All right. So nowadays, Alhamdulillah, we have the cell culture, the tissue culture technology. At the same time, also like in Malaysia now, it's getting more popular to work with zebrafish to study certain biological condition or biological physiological experiments is done on zebrafish in Malaysia. And also, of course, there's another type of animal called the worm. I forgot the name of the worms. It's one type of worm that has been widely studied, especially in aging issue of aging. Yeah, so that is also quite common and replace the use of mammalian and rodents animal in the laboratory. Right? Yeah, but our topic today is on cell culture. So cell culture helps a lot in addressing the issue of reducing animal use, right? Right. Right. So yeah, why we do need cell culture? Number one, there has been applied as major tools in cellular and molecular biology, studying genetics and molecular genetics, and also cellular biology. I think has benefit a lot from the cell culture technology. All right, and then cell culture provide an excellent model system for studying the normal physiology and biochemistry of cells. For example, in the study of the metabolic condition, biochemical condition, and also very popular in the research on aging. I think in Malaysia we have very good collaborative work on aging research in University Science Malaysia, my alma mater, in University Science Malaysia and National University of Malaysia. They have collaboration in focusing on the study of aging. That's also apply the cell culture technology. And then the study of the effect of drugs and toxic compounds on cell, directly on cell. So we use cell culture technology to do that. Before we proceed to study on animals and so on at the bigger level, we study on cells first. All right. This is very common, especially when we have a compound, a new compound for any treatment. So we test on cells to study the toxicity, right? So I think anyone who work with cell culture is quite would be very familiar with the term MTT, right? The MTT is assay. Now, before we do any treatment, we do the MTT assay first to identify the CD50 of the compound on the cells. That is, I think, very, very common. And then the study of mutagenesis, what is the mutation, how certain compound would induce mutagenesis, all right? And then the carcinogenesis also, to study whether certain compound can induce cancer in certain cells, right? So yeah, we grow the cell doing cell culture. Right? Also very common in drug screening and development, all right? And then also for large scale manufacturing of biological compound, for example, vaccine, right? We know, I think it's been very popular recently in these few days that the vaccine for COVID-19, the potential COVID-19 vaccine is almost ready. Inshallah, in a few months we would have the vaccine. Inshallah, yeah. And I heard that Indonesia and Malaysia has already sort of like have the agreement to buy some of the vaccine from China and also some from the European countries also. All right, so we will see what would happen with the vaccine. So the vaccine development also require cell culture technology, right? The mRNA vaccine especially, we have to grow treat the cells with the mRNA of COVID-19 and then to see whether the cell would produce the compound that is specific for COVID-19. The same thing we do with other viruses especially, right? And then to the production of the therapeutic protein, all right? And then production of antibody, this is very common. And this is also one of the field that I'm very interested in production of monoclonal antibody for certain cancer, right? And there are actually so many monoclonal antibody in the market that has been produced thanks to the cell culture technology. We can also produce hormone in the laboratories using cell culture technology. Yeah, so again, some of the advantages of cell culture. So researchers can have absolute control of the physical environment of the cell culture if we grow it in the lab. We can maintain the homogeneity of the sample. Less compound is needed than in animal model, all right? And we can ensure or sort of ensure consistency and reproducibility of results, right? Because we maintain all of the physical environment and the condition, we can make the production line very stable, right? There are some of these disadvantages. Number one is it may be hard, certain cells may be hard to maintain because of the slow growth and very high possibility of mutation, high rate of mutation. So some of the cell are very hard to maintain. All right, and only grow a small amount of tissue, and of course, one of the main issue is the cost lah. The cost can be very high to maintain the cell culture laboratory. All right, and also de-differentiation. The cell changes over time after a certain number of passage. And this also relates to the instability and aneuploidy that, as I mentioned earlier, seen very commonly in HeLa cell, right? Aneuploidy, which is changes in the number of the chromosomes, all right? Which is which means that the cell has actually changed from its original state, right? Okay, so these are different type of animal cell culture. All right, of course we usually start, we want to have a new cell line, so we start from the organ, we take from the individual subjects, we do the cell fragmentation, we do enzyme digestion of cell fragment with enzyme, for example trypsin or collagenase, and then we have the single cell, and then we culture the single cell in culture media. This is what we call as primary cell culture. And then we do, if we proceed with frequent subculturing, around 70 to 80 times of primary cultured cell in the culture media, this is what we call as established cell culture, right? And then if we do after more than 80 subculturing, and then they may be introduced, there are some alteration in the cell division characteristic has been introduced, and change in chromosomal number, and cells actually acquire indefinite growth property. This is what we call as transformed cell culture. This is what we seen in HeLa cells, because so many times it has been subcultured, it gains certain ability that is not normal, totally abnormal, right? So it can acquire indefinite growth property, right? It can grow indefinitely, right? So that is actually a transformed cell culture. Right? Primary cell culture is derived directly from excised tissue and cultured either as outgrowth of excised tissue in a culture or dissociation into single cell by enzymatic digestion or mechanical dispersion. The advantages of using primary cell culture is usually it retains many of the differentiated characteristic of the cell in vivo. It still have the characteristic of the normal cell in vivo. If we limit the use of the primary cultures. And then the disadvantage is it is initially heterogeneous, but later become dominated by fibroblasts. This is also a problem when we work with primary cell culture. It could be dominated by fibroblasts after a while. All right. And then the preparation of primary culture is labor intensive, taking from the organ and then doing the downstream work. It's usually labor intensive, and it requires someone who is very good in handling the cell culture, right? And then it can be maintained in vitro, but only for a limited period of time. It's not indefinite, right? And for the continuous culture is derived from subculture or passage or transfer or primary culture, right? And the subculture is the process of dispersion or reculture of the cells after they have increased to occupy all the available substrate in the culture. So this is the when we have the one primary culture, we do subculturing to maintain, to make sure that the cell is not too crowded in the flask, right? And then it usually comprise of single cell type. If you work with cervical cancer cell, usually that is the only cell that we work with in the entire flask. It can be serially propagated in a culture of several passages, and there are two types of continuous culture. Number one is cell lines, and number two is continuous cell lines. I think I would not delve too much on the technique of the cell culturing, because also they are part of bioethics and also Islamic perspective that I think supposed to present also or to share. So continuous cell lines can be propagated indefinitely for many cycle of differentiation, right? And generally have this ability because they have been transformed, right? This is very common to have continuous cell line for tumor cells, because tumor cell has that ability to propagate over many cell cycles, right? Mitotic cell cycles. And then a cell that has been transformed using the viral oncogenes. We can also manipulate cell lines by infecting them with viruses, and it change the viral genes, viral oncogene, and it changed the cells behavior to become sort of like the cancer cells. Or we can also do chemical treatment to give the cell the ability to become continuous cell line. Right? the disadvantage of having retained very little of the original in vivo characteristic. Again, this is an issue that the continuous cell line have very little properties of the original in vivo characteristic of the cells that we are studying. It's not a normal cell per se. Right. And then another new technology and very now very popular everywhere, I think, is the stem cell. Right. Instead of using a cancer cell line, we use the stem cell that has the ability to differentiate over many cell cycle rounds, and also cells that have the ability to develop into different type of cells. Now, this is a very primordial cell that very early at the developmental stage, and has the ability to grow into many types of differentiated cell, depending on what they call it, what stimulation that we give to the cells. Right? What are the environment that we provide to the stem cell? Right? A stem cell is a cell with the unique ability to develop into specialized cell type in the body. Right? In the future, they may be used to replace cells and tissues that have been damaged or lost due to disease. So stem cell is a cell that at early stage of the development, and now we take usually from blastocyst, okay, from the embryonic cell, and then grow them into cardiac cell or erythrocyte. It can be a fat cell, whatever cell that we want to them to differentiate into, depending on the simulation. We have one group in IIUM trying to work on reviving the rhino. I mean, the Peninsular Malayan white rhino has sadly extinct, very declared extinct in Malaysia very recently. We don't have any more white rhino so what this group is trying to do is to use the stem cell technology, and then use they take the cells from adult rhino, isolate them, and then grow in the laboratory, and then try to treat them and grow them into an embryonic cell, grow them into ovum and also sperm, and then try to do in vitro fertilization in the lab, so that they can have another rhino. But I don't know whether this would be successful. That is one of the very big project that they are trying to do. Yeah, but it is still very early stage. We don't know whether it will be successful. Never done before. All right. And in Australia, actually, there are also one group that is trying to do using the stem cells technology to introduce the Tasmanian tiger. They want to have Tasmanian tiger, and then using the stem cell technology from the cell that has been preserved from the late 19th century, right? So okay, that's about cell culture. And now we move on genetic engineering, right? The genetic engineering is the process of using recombinant DNA or rDNA technology to alter the genetic makeup of organism, right? So when we refer to genetic engineering, even though it may also refer to the use of RNA, for example, not only DNA but also RNA, right? So traditionally, humans have manipulated genomes indirectly by controlling breeding and selecting offspring with desired traits. So we actually had done indirect genetic engineering by the farmers, by choosing certain traits, and then try to make sure that only those kind of traits being passed down to the generation of the animal, for example, or plants, right? For example, if we talk about the our paddy again, our paddy is actually, and also the wheat, it's actually very, very different from the original wild plant. That is the origin of paddy, right? So if we study the genome, it's actually very different, right? So we have introduced the sort of mutation, the good mutation that we want, so that our paddy become more, lebih gemuk, lebih subur, kan, lebih cepat panennya, kan. Jadi that is through indirect genetic engineering, right? Through crossbreeding and so on and so forth. But when we talk about the more modern genetic engineering technology, we directly go to the DNA and change the DNA, right? So in medicine, genetic engineering has been used to mass produce insulin, for example, human growth hormones, follistatin or for treating infertility, human albumin, monoclonal antibodies again, anti-hemolytic factors, vaccine, and many other drugs. We use, we in the cell, we change the DNA of the cell and then produce certain proteins that we use for therapeutic industries, for example. The earliest actually is the insulin, right? We use the originally the sequence of insulin, the genetic sequence that code for insulin from cow, for example, and that from cow cell, and then but we change so that now instead, because there are some differences between the insulin of cow and insulin of human, right? So the cell from cow is changed using the DNA sequence is changed, introduce mutations so that the new insulin sequence now is very exactly the same as the one in human, so that the cell now produce human insulin instead of cow insulin. Okay, so changing directly the DNA at the DNA level, that is what the recombinant DNA technology has allowed us to do nowadays. So in research, the organisms are genetically engineered to discover the function of certain gene. The transgenic mice technology, for example, I was in Sweden during in 2008 when the scientists that introduced the transgenic technology, Oliver Smith is received his Nobel Prize. So alhamdulillah, I attended his lecture and learned a lot about the transgenic, how they introduce this recombinant technology to produce transgenic animal, mice, and then that help a lot to understand the function of certain gene. If we introduce, we change certain gene, what happened to the mice, right? So with that, we understand what is the function. Indirectly, we study, or help us to understand what is the function of the genes, right? So there of course there are many issues discussed on this technology. All right. So all right. So other application for, oh, actually this slide should be a bit earlier before this genetic engineering. Forgive me for that mistake. Right. So again, back to the cell culture technology. So the application of cell culture or mammalian or animal cell culture technology, a lot of them used as animal models. But the one that I want to highlight is the monoclonal antibody, because this is the one that been trying to focus on. Even though now I'm not directly working with this anymore, but this is the one that I used to work on or focus on the production of a vaccine and monoclonal antibodies, right? Okay, so we actually apply the cell culture technology for the production of monoclonal antibodies, right? So originally, how we do antibodies, for example, antibodies against certain poison, for example, or certain snake poison, for example. Yeah? So we usually what is done is we take the poison or the venom from the snake and then inject into horse or into sheep, and then allow the horse and the sheep to produce the antibody against that. So in doing that, we actually another issue is giving harm to the animal. Of course, the horse introduced with the poison will produce the antibody, but at the same time, the horse will also be harmed. All right. So, and also another issue is the antibody that the horse is producing may not be may not be similar, will not be exactly the same as the antibody in human. Of course, there are differences in the antibody structure between human and horses, so that when we want to use this antibody to treat the people who have been bitten by the snake, the body of the people will also react to the horse antibody. It may also causes the immunological response that is not desirable. So nowadays we have the monoclonal antibody technology. Instead of using other mammals to produce the antibody, we can grow the antibody in the laboratory, in vitro, using the cell culture technology. All right, and then with this technology also allow us to produce monoclonal antibody, one specific antibody for whatever function or application that we want to do. Instead of when we use the other mammalian like horses or in sheep, the type of antibody that is produced is polyclonal antibody. All right, the various type of antibodies targeting the same poison. Okay, but in the cell culture technology, we produce monoclonal antibody, one type of antibody, which is very specific, right? And this has been used, right? So in the monoclonal antibody technology, what we do is we use the mouse challenge with the antigen. For example, the snake poison, for example, snake venom, challenge the mice, and then we harvest the spleen cell from the mice, and then we fuse the spleen cell with myeloma cell. Right? We diffuse, and then we do the fusing, and then this will produce what the so-called hybridomas. Right? Because myeloma cell is actually a type of cancer cell that has the ability to fuse with normal spleen cell, right? So we have hybridomas, and then we do several rounds of cell culture in a very specific medium that we call het medium, to ensure that only cells that have the characteristic of the hybridomas will grow, and then from here we harvest the monoclonal antibody, right? And by using this technology, there are so many drugs, or there's so many treatment that we have today actually based on this technology, right? There are so many diseases that now treated using the monoclonal antibody. Any one of those, any of the drug that is marked in the end of the name means that it's a monoclonal antibody. All right. One of the I think some of the popular one is Avastin. I think medical doctor will know this. It's actually a humanized monoclonal antibody for the treatment of colorectal cancer. All right. Also, for non-Hodgkin lymphoma, we have Bexa. We have also Campath. for the treatment of chronic lymphocytic leukemia, a disease that is very common in European countries. We also have, I think this is very common also, very popular, Herceptin for targeted therapy of human breast cancer. All right. So these are the example of the antibodies that is produced using the cell culture technology. All right. Okay, we go to bioethical issues on cell culture and genetic engineering. I will just give a very broad or very surface introduction on the bioethical, general bioethical issues on cell culture and genetic engineering. Number one, the issue that usually being discussed, number one is the ownership of human derived cell lines and stem cell. Who actually owns the cells? And do we have to recognize from where, from whom the cell line, especially when we talk about the cells that is taken from human, right? As I mentioned earlier, there's even a movie, the movie in Indonesia, I forgot the term, not bioscope. Bioscope is the cinema, right? How about movie? Also called movie in
film or
film.
film.
Yeah. There's a film about Hila, about Henrietta Lacks, the immortal life of Henrietta Lacks. Very interesting movie about how actually the way that the doctor in John Hopkins University took the cells is very unethical if we consider our current understanding of bioethics, right? They don't recognize the individual, and then there's no informed consent before they take and they grow the cell. And also the issue of selling the cell also become very hotly debated. All right. And then not only the cell or the stem cell, ownership of the derived data, whatever information that we get from the research that that come out from the experiment using the cell line, actually who owns those data? Is it the country or the laboratory that originally took the cells, or the researcher, the end researcher that actually do the research, right? Do we have to recognize also the researcher that provide the cell lines, or is just the end researchers? So that is also another issue. Who owns the data, right? Another issue is actually being discussed and being debated is the possibility of production or invention of the so-called chimeras. This technology allows, cell culture technology allow us to change part of the DNA using the genetic engineering, right? We change, or we can now input using the recombinant cloning technology, so we can insert certain gene, for example, from mice into the cell of a human, that would produce the so-called chimeras. The cell that is actually carrying a gene from two different not related species. This is quite common also when we see the monoclonal antibody production just now. So we use the spleen cell from the mice, and then we use the multiple myeloma cell, which is a human cell. So would that, the hybridomas, is it a chimera? Because now it carries, it is one cell, it carry cells from two different species, right? Human and mice. So is it ethical to do that, or what is the issues? ethical issues when we do that. Right? That is at cell level. But what if the researcher grow this cell into an organism? Right? So would that causes a problem or bioethical problem in the future? Right? There's also issue that has been hotly debated. Another thing is, of course, the issue about human embryonic stem cell. One of the biggest issue about why human embryonic stem. When you talk about stem cell, there's two type of stem cell technology. One is human or embryonic stem cell. Number two is pluripotent stem cell, which is embryonic stem cell we take from the embryon, right, from early stage of the gestation. While pluripotent usually we take from the mature cells and then we make it into a stem cell, right? So for the pluripotent stem cell, I think the issue is not that hotly debated, but for the human, especially human embryonic stem cell, is very hotly debated, even in the Western countries, even in the US, because to do the in the process of making embryonic stem cell, the embryo itself would be destroyed in the end, right? Does it means we are killing a human when we take the human embryonic stem cell and growing it in the laboratory? Yeah, that is also a bioethical issues that is being discussed in general. So in many countries, human embryonic stem cell is not allowed. I think in the US it's not allowed, but in some of the European countries it's allowed. Yeah. Another issue is about biobanking. All right, the biobanking, how to maintain, it's also an issue. All right, because all of the cell line technology, we do need biobanking. Right? How about how do we ensure that the biobanking is always functioning and always being done in an ethical issue? How do we monitor all the biobanks? All right. How do we monitor that? For example, if we have biobank that preserve sperm or ovum from individuals, the cells that is involved for the reproduction, how do we ensure that they don't mix and then they don't just give these cells to anyone, or try or to ensure that the biobanks does not cells the cells unethically, right? Another issue is about dual. This is another new issue that's been raised, I think, until the level of United Nations or World Health Organization, is on the dual use of research of concern, right? This technology allows us to do a lot of things, right? For example, we have the cell culture, and by cell culture technology, we can grow, we can do it for a good cause, for the research that has been allowed to be done in our institution. But some of the people may be able to do other dangerous thing with the cell. For example, like if you have the cell lines or cells, people can for example, grow certain dangerous virus in the cell. How do we make sure that this does not happen? All right. So the dual use research of concern is being discussed, and in Malaysia we have set up committees to ensure that this is being monitored, this is being audited in all of the university that we allow to do cell culture technology. All right, so ensure that the researcher will only do the research that they have been allowed to do, and they don't do anything else. All right. Yeah. Biosafety and biosecurity is one of the issue when we talk about again that issue that we discussed. Um, so so when we have a cell culture laboratory, we have to always ensure the biosafety and biosecurity of our facility. All right. So we must ensure the safety of all of the personnel who work in the laboratory. We must ensure the safety of the community and society in the surrounding area of the laboratory. Right? We must also ensure the safety of the environment. Right? So all these issue must be catered before we set up a cell culture laboratory. Right? Also, we have to ensure security. Right? Because security refers to the the you know, the people or the invasion protect from outside threat. When we say biosafety, it means we protect the um, we protect someone or the community from the specimen. But this one is how we protect from outside threat, right? How do we ensure the security of the personnel, right? For example, for example, you work on production of COVID-19 vaccine, you will be a very, very valuable asset to the country. So they may pose some security to you. People may want to abduct you or want to kidnap or some things like that, right? How do we ensure the security of the personnel, for example? How do we ensure the security of the facility? If you do some work on cell culture and there are some people in the community that oppose, that is extremely opposed to what you do, and they may pose threat to your laboratory, want to bomb or want to destroy the laboratory. How we protect the facility? That is the security. So we have, before we do the build the facility, we have to ensure the protection or security of the facility. And then how do we ensure the security of the sample to ensure that no one is taking out the sample from the laboratory? How do we make sure that our students, our postgraduate student, our researcher does not sell the specimen to outsiders, right? So that also have to be taken into account before we set up our cell culture laboratory, right? Right. It becomes more important if we deal with cell culture and involve the treatment or with certain viruses, right? How do we ensure that the sample will remain in the laboratory and not being taken out without any authority, right? So proper risk assessment is very, very crucial, very important before setting up the facility, and the faculty or the institution must have a guideline on prevention, mitigation, and response to any problem, any situation involving the laboratory. So in IIUM, in our university, and I think many of the other universities in Malaysia, we are being monitored by the Ministry of Science and Technology. If we have this facility, if we have a cell culture laboratory, we must register with the Institutional Biosafety and Biosecurity Committee, and this Biosafety and Biosecurity Committee would audit all of the cell culture laboratory to ensure the biosafety and the biosecurity of the lab. So I think it's very important to have the institutional biosafety and biosecurity committee at institutional level, all right? So that we know all of the researchers who do the research in cell culture. We know what they do and what manipulation do they do to the cells, all right? And we know all where does the all the cell is located, all right? So that we have a registry of that facility, right? Okay, so we move on to I think the last part of the discussion today on the Islamic perspective of cell culture and genetic engineering. Let's go to the basic. I must admit that I'm not a Sharia person. I'm a scientist. My knowledge on Sharia is very limited. Even though in the past we do have a bioethicist who is actually very well trained on Sharia. My colleague that I have worked together in studying issues of contemporary scientific technology, bioscience, especially in life science and biotechnology from Islamic perspective. But I myself has to be admit that I'm not a fuqaha. I'm not very well trained in coming up with Sharia or fatwa, right? But you know the basic that I learned from my work with the people in Sharia. In Islam, we have three basic type of hukum, right? When we talk about hukum, we have hukum syariah, we have hukum akal, and we have hukum adat. And these three is recognized in Islam, right? And of course, the number one and the most important in Islam is hukum syariah, which is a revealed law of that Allah Subhanahu wa Taala has passed down or has said to every one of us, the Muslims, right? So in hukum syariah, of course, we have the three most important sources, which is the Quran, the Hadith and Sunnah, the recorded speech, and also the acts of our Prophet Rasulullah Sallallahu Alaihi Wasallam, and also the consensus, the issues that has been solved due with the consensus among the early generation of fuqaha, the Sahaba, the Tabi'in, the Tabi'in, also the early generation of the fuqaha, right? So we have the records of that. So in any issue that we face today, number one, we must refer to all of these resource, all right? And then if we don't have a solid answer, we still have to discuss, all right? So we have to refer to the fatwa that has been done based on qiyas or analogy, and the maslaha mursala, public interest, the istihsan or juristic preferences, adat and oath, customary practice, istihsab, presumption of continuity, sadul darai, which is blocking of mean. This is the basic or the methodology of coming out with fatwa. For any issues, like for in Malaysia, I think we may have differences in terms of the arrangement or the management of the Islamic or religious departments. Like in Malaysia, we have I think you all know in Malaysia we have 9 states. We have 14 states and 9 states with sultan. So in those states with sultan, the sultan is the highest authority in any issues regarding Islamic issues, and the sultan usually would have the mufti. So in all of the 9 states, we have the mufti. So any issues, we have to refer fatwa from the mufti. You have to bring the issue to the mufti, and it is the mufti who would come out with the fatwa or the decision for the state. And for the rest of the 5 states, they don't have mufti, but they would have the federal they would have the fatwa from the federal level, right? So that is how it is in Malaysia. So we must always say that, you know, when we ask about fatwa, we have to ask the mufti and not any other individual. All right. And also in Islam, we recognize or we establish that Islam also take account the akal. You know, when coming out with hukum, hukum akal is also recognized in Islam. This is the decision or hukum that is based on evidence-based observation and deduction, scientific and mathematical analysis, understanding of sunnatullah, and everything that happens, it is within the law of Allah that Allah has set in everything and anything in the world. The law that we call as sunnatullah. Now, we as a scientist, we only study sunnatullah. All right. So yeah, and also in Islam, we have tafsil birra'i, meaning that we interpret based on explanation based on reasoning. All right. Islam recognize reasoning also to come out with the Sharia decision. And also historic narrative. We also can take into account the historic narrative of certain issues to come out with the rule or guideline. Adat is also taken into account in Islam and recognized. And this refers to good practices and customs. There are certain differences between different places in terms of practices and custom, and then they are allowed to be taken into account when we talk about Islamic law, as long as they do not conflict with the divine law, with the Sharia, right? So we always go back to the Sharia as the number one references to come up with the decision, with any decision, all right? So this is, I will present only three. There's one book written by my colleague, Dr. Abdul Razak Hashi. He's a very good fuqaha, I think. In IIUM, he is one of the main references with regard to contemporary issues in Islam. And he has produced a book called Bioethics, a very good book. So far, this book has already been sold out in our university. Everyone wants this book, and hopefully, I've told Dr. Razak, maybe in the future we will have more printing of this book, so that many more people could benefit and learn from his expertise. A very good book, inshallah. So in this book, he actually gave 12 fundamental principles of Islamic ethics. But for today's sake, for the sake of time, I will only present three, the main fundamental principle that is presented in this book. Right. So number one, Islam holds the world view that human is trustee and hold responsibility of being the vicegerent or Khalifa on earth, as Allah Subhanahu wa Taala said in Surah Al-Baqarah, chapter 2, verse number 30. وَإِذْ قَالَ رَبُّكَ لِلْمَلَائِكَةِ إِنِّي جَاعِلٌ فِي الْأَرْضِ خَلِيفَةً قَالُوا أَتَجْعَلُ فِيهَا مَن يُفْسِدُ فِيهَا وَيَفْسِقُ الدِّمَاءَ وَنَحْنُ نُسَبِّحُ بِحَمْدِكَ وَنُقَدِّسُ لَكَ قَالَ إِنِّي أَعْلَمُ مَا لَا تَعْلَمُونَ. Just think, when your Lord said to the angels, lo, I am about to place a vicegerent or Khalifa on earth, they said, you will place on it one who will spread mischief and shed blood, while we, the Malaika, celebrate your glory and extol your holiness. Allah subhanahu wa ta'ala said, surely I know what you do not know. Allah knows better why He put us on this earth as Khalifa. Right? We have the duty and the responsibility to ensure that whatever we do, especially we as Muslim, or if we Muslim are the ones who have the authority over the land, to ensure that everything that is being done under our authority is in accordance to the Sharia, is according to what Allah Subhanahu wa Taala has put on us to manage and to make sure being done properly. So the second principle, fundamental principle, is resources are subjected to human use, yet human is morally bound to properly manage according to Allah Subhanahu wa Taala's order and good consciousness. Right? As Allah Subhanahu wa Taala said, kullan numid haula'i wa haula'i min ata'i rabbik wa ma kana ata'u rabbika mahpura. We provide both the former and the latter from the bounty of your Lord, and the bounty of your Lord can never be withheld. All right? Allah Subhanahu wa Taala give us everything in this world for us to take benefit, but it must be done in good moral bound and must be managed properly. And Allah Subhanahu wa Taala also said, "Inna fi khalqis samawati wal ard wa ikhtilafil layli wan nahar wal fulki allati tajri fil bahri bima yanfa'un nasa wa ma anzalallahu minas sama'i mimma'in fa ahya bihil ard ba'da mautiha wa batha fiha min kulli dabbatin wa tasrifir riyahi was-sahabil musakhkhari baina as-sama'i wal ardi la ayatin liqaumin yaqilun. Indeed in the creation of the heaven and the earth, the alternation of the night and day, the ship that sail to the sea for the benefit of humanity, the rain set down by Allah Subhanahu wa Taala from the sky, reviving the earth from its death, the scattering of all kinds of creatures throughout, the shifting, everything is for human to reflect and to learn and to take responsibility. Right? And also the third principle is ethical actions are conducts that are in line with the inborn character of human, the fitrah. Everyone want to, a good human being, want to do things according to the ethics. All right, ethics is in line with our fitrah. Allah Subhanahu wa Taala said, "Faqim wajhaka lid-din hanifa fitratallahi allati fatara an-nasa 'alayha la tabdila lil-khalqillah dhalika ad-dinul qayyimah walakin aktharan nasi la ya'lamun." So be steadfast in the faith, in all uprightness, O Prophet, which is the natural way of Allah, which He has instilled in all people. Let there be no change in this creation of Allah. There is the straight way, but most people do not know. The fitrah, being good, being ethical is part of our fitrah. We want to preserve that, ensure that in our doing, in daily life, in whatever we want to do as a researcher and as a scientist. Allah also said, "Ya ayuhan nas, inna khalaqnakum min dhakarin wa unsa wa ja'alnakum syu'uban wa qabail lita'arafu. Inna akramakum indallahi atqakum, innallaha alimun khabir." O humanity, indeed we created from a male and female and made you into people and tribes so that you may know one another. Surely the most noble of you in the sight of Allah is the most righteous among you. Allah is truly all knowing, all aware. So everything that we want to do is according to the fitrah, and we want to strive to do to become the righteous servant of Allah subhanahu wa ta'ala by ensuring that whatever we want to do, whatever research what we want to embark on is in accordance to his Sharia and his Islamic ethic that he has guided us on. All right. So in achieving the Sharia, to ensure that whatever we do is in accordance to Islam, we have this is what has been outlined by the great scholar of Islam Al-Ghazali about the Maqasid al-Sharia. And this is, I think, in Malaysia we have adopted the Maqasid al-Sharia to organize or to guide any efforts or any work we do in our academia. All right. So in Maqasid al-Sharia, or objective of Islamic moral law, we always, whatever issue we have, we always return to the Quran and Sunnah as the basis of the Sharia. We also take it into account, the Maqasid al-Sharia, and as the overriding strategy or overriding principles, right? So in the Maqasid al-Sharia, we ensure that this 5 objective of Islamic moral law is always fulfilled at all time. Number 1 is what we call as Hifzuddin, right? So whatever we do, we must ensure that kita pelihara agama, all right? Islam, pemeliharaan agama di kalangan umat Islam itu adalah sangat penting, dan that is the first thing, right? We must ensure that whatever we do not threat our own religion, right? Pemeliharaan agama adalah satu yang sangat penting, dan yang pertama dalam maqasid syariah. And number 2 is hifzun nafs, pemeliharaan jiwa. All right, we must ensure that life is taken very important. We must protect human life all time. This is the very basic principle, very important principle. Number 3 is hifzul aql. All right, protection of the knowledge, sanity and knowledge and intelligence, right? And then hifzul nasl or pemeliharaan keturunan or protection of offspring, and then hifzul mal, pemeliharaan harta. Right? This is the 5 basic principle of Maqasid Sharia. And when we build the Islamic bioethics, we build based on the Maqasid Sharia, right? And in any issue, we always refer to this to discuss and to provide the guideline. All right. So I think this is the last slide. Sorry, this is second last slide. So in institution, I think I'm not sure. I think in Nusila you must have the vertical committee. Yeah. So it's very important. It has the responsibility to ensure good and responsible research and practice conduct, which is rest on the shoulder of every researchers involved. We must put that the responsibility on all of our researchers, right? That they are responsible to ensure good and responsible research and practice. Okay. However, institution and authority must take active efforts to monitor all research conduct related to cell and tissue culture and genetic engineering. Sorry for the mistake. Right. So yes, we put the responsibility on everyone, every researchers in our institution but the institution and the authority must monitor, must audit every activities that is taken or being done in the laboratory, especially those related to cell and tissue culture and genetic engineering. All right. We must set up and empower, provide empowerment of institutional bioethics committee. So this bioethics committee must be empowered. So if they, for example, they discover any misconduct in the lab, this bioethics committee has the authority to close down laboratories. All right. So that is the very important also, so that everyone is always being responsible. All right. Okay. And then also it's very important. We should train. I think Unisula as Islamic University, also IIUM as Islamic University, we do need to train and employ expert in research bioethics. We have to have expert in bioethics that understand also the Sharia and the Islamic perspective of of the issues regarding to work in life science and biotechnology. I think this is one of the field that I think we need to fulfill still as our fardu kifayah, I think. So in IUM, we have Dr. Abdul Razak as our bioethicist, Islamic bioethics. We have also another professor in this field, but we realize that we need more. This field, I think, university like us, Unisula and IIUM should collaborate and should be more empowered. All right. So I think this is one of the field that we can collaborate further in the future, so that we fulfill this fardu kifayah for our ummah, inshallah. So all related activities must be vetted, must be recorded, must be registered, all right, before embarking. So before any work on cell culture and genetic engineering is done, the proposal for those work must be passed, must be submitted to the committee, and the committee must vet and must read properly, and then must decide whether the research is allowed or not. All right, and then the progress of the research must be monitored effectively. So even though we have given the approval for the research to be done, the research will still be monitored to make sure that all of the researchers do the work according to what they have told the university, and they not do anything else. All right. So ensure that they don't do the dual research. Right. So yeah. So we need national and institutional approach to ensure that all work in cell culture and genetic engineering is done properly. Right. The institution must have clear guideline and protocol. Right. When we have cell culture laboratory, we have genetic engineering, we must have clear guideline and protocol. Yeah. So in IIUM, we had only very recently, in 2019, we published the guideline on biosafety and biosecurity, and in that we also include all the laboratories. So all this laboratory must be done according to that guideline. Right? It's very important for us to have that. We must set institutional and national ethics, research ethics committee. So in IIUM, we have our IIUM institutional research ethics committee, and at national level also we have national Malaysian research ethics committee. So all of the research all of research proposal must be submitted to both, and it must be approved before we are allowed to do our research. All right. So it's very important to ensure that all the research proposal is being read and vetted. All right. Active involvement of community and research and religious group representative must be, we must have it in the research committee. Okay. So in our research committee, we have a lawyer, we have judge, we also have a representative of the community, leaders of NGO, certain NGO, and also we have the representative from religious group, from the, for example, like IIUM Pahang, is located in the state of Pahang. So we have involvement from the Majlis Ugama Islam Negeri Pahang as one of the representative for our research committee, so that they can also advise us on issues regarding the scientific proposal from the Islamic view, and also community view, society view of the society. And we, the institution must ensure appropriate facility covering responsible research, biosafety, and biosecurity principle. Right. So yeah, it's the duty of the institution, the faculty, and the university to provide the appropriate facility lah. Because when we work with cell culture, we know that it is quite expensive to maintain. But when we decide that we want to have cell culture technology, yeah, the facility must be appropriate. We must have a registry of all approved research proposal. All right. So all of the record must be kept. All right. And any amendment and completed research should be recorded. So university must have all of the record lah. All right. And then yeah, the record must have the proper record on laboratory, personnel who work on this field, type of the cell that has been worked on, and then all of the manipulation has been done on the cells. Right. Access to the facility must also be recorded. Right. I've seen laboratories that have a very advanced technology. All of the record is digitalized. All of the record are automated, right, in a computer, very safe. But also I have been to a laboratory which is, everything is done manually. All of the access to the laboratory is done on the logbook. It doesn't matter which approach we can afford and what approach we want to choose. But as long as we have proper record, then I think that is vital, right? Certification, authorization, and awareness and repercussion on transgress must be always, being told to the staff, right? So we have must always remind our researchers, our staff, to remind them on repercussion on transgress, neglect, and any responsible action. They must always be reminded. All right. So I think this is all for me. Sorry for taking more time. Yeah, I'm open for any question. Back to Dr. Fikri.
Okay, thank you very much, Mr. Arifin, for the very great lecture. Okay, it's time for the question. So every student or lecturer that join this lecture can ask the questions. The question can be sent by IIUM chat or directly by raise hand. Okay, there is one question from Dr. Hadi Sarosa to Mr. Arifin.
Yeah.
In Indonesia, apa keuntungan dan kerugian pengembangan vaksin melalui cell culture technology? Oke, baru ada satu pertanyaan, Mr. Arifin. So, please.
Jadi pertanyaannya berkenaan dengan benefit ya, benefit.
Benefit and yeah.
Of course, bila kita berbicara tentang vaksin, tujuan vaksin itu sendiri adalah untuk memberikan potensi perlindungan kepada komuniti kita daripada penyakit-penyakit yang daripada penyakit-penyakit infectious disease, all right, especially. So nowadays, I think when we talk about the development of vaccines, it's very popular for us to apply the cell culture technology. For example, like the production of COVID-19 vaccine, and also I think the influenza vaccine. These are the two diseases that cause emerging pandemic threat recently. So setting up of the vaccine or in the production of the vaccine, we apply the cell culture technology. One of the reason why, one of the benefit of why we do cell culture technology for this vaccine production. Number one, as I mentioned earlier, in the past or conventionally, people use animal to produce vaccine, right? For example, like for tuberculosis, the work by I forgot the name of the researcher, the French researcher that produced the vaccine, right? The tuberculosis vaccine. They use cow, right? The cowpox vaccine to treat, right? So they use in animal, but there are issues when we use animals. Of course, the use of a lot of animals is always an issue. But when we do the cell culture, we can avoid from using animals, right? Or we can reduce the use of animal, and it is more convenient to use cell culture. And for like for COVID-19 vaccine, the COVID-19 vaccine, especially the one that is being developed by Pfizer or being commercialized by Pfizer, is actually developed by two researchers from Turkey, right? Turkish researcher that live in Germany. I think husband and wife, and the husband and wife actually are Muslim. They are both Muslim. They have been working on vaccine production many years, but now they give the Pfizer to commercialize. All right. So when developing mRNA vaccine, because like COVID-19 coronavirus is RNA virus, so it's very difficult to develop vaccine using the conventional technology because it's an RNA virus. So using the cell culture technology, we can inject the mRNA itself so that the cell will produce the protein, and this protein can be used as the vaccine, right? So yeah, so it allows us to produce vaccine for virus, for example, especially for virus. I think the only way, the best way to do it is through cell culture and genetic engineering technology. I hope that answer.
Yeah. Okay, thank you very much, Mr. Arifin.
Yeah.
For the other questions from Mr. Setio Trisnati, our dean,
Yeah.
He has a question: bagaimana kebijakan Malaysia untuk laboratorium medik untuk riset dan produk stem cell tentang perizinan hak paten?
Terima kasih banyak kepada Bapak Dekan. I actually have to admit I don't have a very good answer to this because for the stem cell technology untuk hak patent, very important question, but I'm not sure whether I have a good answer for this. Let me allow me Pak Dekan untuk berfikir bagaimana untuk menjawab pertanyaan ini. I think for any work, if the research is funded through a national funding agency, right? So the hak of the patent is shared between the researcher, the institution, and the government, the funder, right? So it is very clear if the funding is coming from the national resource, but if the funding is from outside and from the collaborative efforts, for example, like collaboration between Malaysian and Indonesian university, the izin for hak patent must be defined before the research is being done. So the Ministry of Education, Higher Education, has reminded us that whatever agreement in terms of hak patent must be defined by an MOA, Memorandum of Agreement, before the research has been done. So, so agreement between the researcher must be defined earlier, and not and not after we get the result. Only we try to fight each other. Then this is very common, lah. If we don't define it earlier, when we come out with something that can be patent, if we don't define it, it may cause problem between the researchers and between the country, and even so may cause some diplomatic problem. So it must be agreed before we do the research. How many percentage? What are the contribution of each of the collaborators? What are the contribution of the funders? So that must be contributed, must be defined. So if for example, the grant come from outside, right? And the researcher is Malaysian, we must define the percentage, the agreement of any of the role and any of the responsibility, as well as the profit from the patenting of the product from the research, right? So I think that's what I know. Mohon maaf kalau jawapannya tidak begitu bagus.
Oke. Karena di Indonesia masih menggunakan bahasa Indonesia masih menjadi problem.
Iya.
Problem. Jadi termasuk perizinan labnya. Labnya kalau di Indonesia lewat Kementerian Kesehatan, kalau di Malaysia kan Kementerian Riset dan Teknologi, ya. Iya,
mostly.
Terus kemudian ada grant-grant tadi, tetapi terbatas pada kelompok tertentu kalau di Indonesia. Kami sebetulnya kan ada lab SCCA tentang stem cell itu yang dikerjakan Profesor Pak Agung itu. Akhir ini saja yang bisa bekerja sama dengan kementerian terkait dengan produk skretum untuk penderita COVID yang berat. Itu kami melakukan itu, Prof. Ya. Terima kasih sharingnya, Prof.
Ya. Terima kasih, Prof.
Thank you, Mr. Setio and Mr. Arifin. Yeah. Next to the other questions, this is question from a student. He asking about dalam perkembangan teknologi kultur sel, pernah ada percobaan yang mencoba membuat organ vital seperti jantung, ginjal, hati, dan sebagainya. Apabila ada, bagaimana potensinya untuk teknologi transplantasi dari organ buatan seperti hal tersebut di masa mendatang?
Ya. Terima kasih Dr. Fikri. Terima kasih juga pada yang bertanya. Sebenarnya ada juga percobaan ataupun attempt untuk membuat organ seperti jantung, especially. I know of one of the research, satu group research di University Lund University di Sweden semasa saya di sana dulu. Memang mereka mencoba untuk mendevelop ataupun menghasilkan jantung, sel tisu jantung yang boleh berdegup. And they have been successful in developing the beating heart tissue, and it gives potential that in the future we can develop very specific organs, heart from this technology. So we don't have to rely on, because jantung very difficult to do transplantation, right? There were attempts in the past to transplant heart from khinzir again, from pigs, but there's a lot of issue, and also it not very successful because it's what we call as transplantation between species. Will always cause rejection. All right. So the best, if you want to do transplantation, is what we call as allogenic transplantation, meaning that we transplant the organ from our own self. For example, we have this technology for skin transplantation, right? Allogenic skin transplantation, in which we transplant skin from different part of the body to the part that, for example, has been burned or has been affected by accident. So if we develop the heart, for example, from the individual, we take the cell from the individual, we develop the stem cell, and then develop towards the functional organ that would help a lot in transplantation. So far, I think the one that I have direct experience is the one in Lund in Sweden. They almost, at least they have reached the level where they can develop a beating heart. But in terms of, because heart is also a very complex organs, which with the chambers and with the injab, with the a lot of complexity in the organ itself. So it may still take a lot of years to actually get a good heart, good cultured heart that can be transplanted into human. But at least they have reached, they have achieved, they have passed one of the biggest challenge, in which to develop a beating cell, because that is one very important criteria of human heart, or any heart for that matter, is that it has it must be beating. It has must beat. So that has been achieved, but I think it may take several years before we have technology to produce functional heart. In terms of ginjal, I have heard about the attempt to produce functional kidney using the stem cell culture technology, but I don't have the detail on that. Yeah, so we will see, inshallah, in the future. But I think this question coming from a student is very, very good indicator that the student is very knowledgeable in this field. So Tanya, Prima, Rizky, Nur Rashid for the question. Right? It shows your concern and your knowledge is very good in this field. All right?
Thank you very much for your explanation, Mr.
Yeah, thank you.
Okay, thank you, Mr. Arifin. Maybe this is the last question, I think, because of time.
Yeah.
There is a question from Hanifah Tiara. Izin bertanya, pasien COVID-19 dengan gejala berat, apakah produk stem cell dapat dijadikan suatu alternatif untuk pembentukan antibodi bagi pasien yang terkena COVID-19? Terima kasih.
Okay. I have to digest the question. I don't know whether I understood the question correctly. Dia bertanya patient COVID-19 yang gejala berat. So when we talk about patient dengan gejala berat ni, maksudnya yang berlaku kerosakan di paru-paru, macam mana tu? Because at the moment we focus on more on production of the vaccine for the COVID-19. So I'm not sure. I'm not sure how I can answer this question. Atau mohon kalau moderator can help me to clarify the question.
Oh, maybe it's about if the question is about antibody, maybe the question is can we use the stem cell or maybe cell culture to produce antibody for COVID-19, I think. Because if like this severe disease, I think antibody is not good anymore because antibody need time to get the.
Yeah. So I think if the patient has already been affected and I'm not sure how the antibody can help, because if the patient has already been infected and already, for example, in the issue of COVID-19, it's the pulmonary system. The lung usually collapse, or the lung has been severely affected, and the patient has lost a lot of potential or volume or potential in the function of the lung. I'm not sure whether how antibody can help on that, because what we need in this kind of patient is the treatment, the reduce of the symptom, or help the patient to recover from the symptom of the COVID-19, right? Because when we are infected by any virus, of course, if the body is good, immune system is good, the immune system will produce antibody against that virus. But I'm not sure how we can help the patient to produce antibody. But when we talk about the vaccine that we want to produce, right, it is to prevent people from getting COVID-19, and how we do it is by introducing the RNA, mRNA of the COVID-19 into the cell, and the cell produce the antibody. So this is before the COVID-19 has affect the individual, to provide the protection. So, but for the patient, I'm not sure. I'm sorry. I'm not sure whether that can be done, because there's actually an attempt in Malaysia. There's an attempt in the beginning of the COVID-19 to collect the blood of the individual who have recovered from COVID-19. So pada waktu awal COVID-19 itu kan, di dalam bulan-bulan kalau di Malaysia, COVID-19 di Malaysia bermula pada bulan Februari. Kes yang pertama yang banyak di Malaysia di bulan Februari tahun 2019. Kemudiannya MCO ataupun Movement Control Order bermula dari March. Jadi dalam bulan Jun itu, selepas daripada Idul Fitri, ada beberapa patient COVID-19 yang dapat recover secara natural kerana mereka sihat dan mereka badannya boleh menghasilkan antibody melawan COVID-19. Kemudiannya ada usaha untuk mendapatkan darah daripada pasien itu supaya nantinya boleh diekstrakkan antibody ya, daripada pasien itu, dan kemudian antibody ini akan diinjek kepada pasien yang sudah terlalu tua untuk menghasilkan antibody, for example. Tapi attempt itu tidak berjaya. Because of course the issue about how much antibody we can extract, because antibody itu tak begitu tinggi kan, ataupun kita tak boleh mengambil darah terlalu banyak daripada seorang individu, apalagi individu yang baru saja sembuh daripada penyakit. Jadi isu in term of volume dan juga in term of allergic reaction disebabkan oleh antibody itu sendiri berkemungkinan akan memberi kesan yang lebih buruk kepada pesakit lah. Jadi usaha itu tidak berjaya lah, tidak diteruskan. Jadi kita masih lagi terpaksa menunggu kepada usaha untuk vaccination lah. Right?
Okay, terima kasih Mr. Arifin atas jawabannya. A very interesting lecture, and then I think time is enough. But I have one question.
Yes.
From me.
Sure.
At the beginning of lecture, Mr. Arifin said that in the cell culture have disadvantages about the differentiations, like culture and passage for many times, so it have a problem on the differentiation. So based on Mr. Arifin experience, how to overcome this problem? Because yes, because when I took my PhD course, my research about SL1 cell, it means the cell line from atrial myocytes. From the fresh cell, I found many cells beating, because my cardiomyocytes have automaticity. That's why the cells still can beating. But after I patient for many times, so only a few cells still beating. The others no.
Yeah.
Yeah.
It's like I've never worked with the heart cell. Yeah. So usually dealing with the cell, we have to know the character of each of the cell line. That's why we have to know how many passage we can do in each of the cell, right? So yeah, usually it depends on the experience and the knowledge based on the very specific cell. So I'm sorry, I don't have the very good answer for the specific cell that you're working with, but I think in general,
maybe for general.
Yeah. So, but in general, I think we usually must communicate with other laboratory that work with the same cell that we do, and then we ask them how many passage that what is the limit number of passage for this kind of cell. So basically, to me, it's like the sunnatullah. Allah has set how many time the cell can divide and still retain its original state. So that is for us, I think, to know and to understand, and to not to transgress. I mean, if we say this cell can only be passed 6 times, then I think we have to accept that, that's the limit, that number, 6 times. So yeah, that's my general comment on that, inshallah.
Okay, thank you very much, Mr. Arifin, for a very good lecture. Okay, maybe for the students, especially for students 2018 that will start the experience, maybe you can consider this technique about the cell culture to take the samples. Because I think the cell cultures maybe looks complicated, but if we do that by ourselves, I think it's not as complicated as we think.
Yeah. Cuma perlu banyak sabar.
Yeah. And of course, we have to prepare many equipment, because the equipment is very expensive at the beginning. But for the next, maybe after we have the equipment, I think to get the sample is very simple and it's very fast as compared to the N50 experiment.
Yeah, that's true.
Okay, thank you very much, Mr. Arifin, for
Thank
very
you.
great lecture. And then I think this lecture is finished, so I will give the time to the Ms. Putri.
Yeah.
Thank you.
Terima kasih, Dr. Arifin and also Dr. Fikri. Please give a warm round of applause and press your reaction button to clap. Thank you, Dr. Arifin. Okay, this has been a very fruitful discussion and very knowledgeable for every one of us. We would like to say our greatest appreciation for Dr. Arifin, PhD. And also, we also have a link for our students and also for lecturer to give feedbacks for today's lecture. And let me tell you the result. 96.4% said that in the future they would like to join if we have if we conduct some international virtual lecture conference like this. So for Dr. Arifin, we would like somewhere in the future we would like to invite you again, because I think the enthusiasm is very clear. And also, yes, and
maybe not only lecture but also research.
Workshops. Yes,
research,
yes, and also research. Yes, joint research between Unisula and UIA, Garden of Knowledge and Virtue.
Inshallah, inshallah, the future.
Yes, and also I have questions from some of our audience here, whether we can have copy of your presentation. I believe you already sent it to me, and I can give it to them. Is that okay?
Yeah, sure. Yeah, you can.
Okay. Thank you very much for today's presentation, for Dr. Fikri, for students, and especially for Dr. Arifin Kaderi, PhD. We would like to close the session by saying Alhamdulillah dan doa penutup majelis. Alhamdulillah. Terima kasih sekali lagi. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Terima kasih semuanya.
Terima kasih Dr. Arifin. Jangan bosan diundang. Okay, we may leave the session.
Alright.
Okay.
Assalamualaikum.
Waalaikumsalam.